Inside the nucleus of every cell, the DNA double helix acts as a stable chemical archive. It is coiled so tightly that the instructions for life are physically shielded from the surrounding environment. Before a cell divides during the S phase, it must generate a second copy of this entire 3 billion base pair sequence. The process requires absolute fidelity. A single error can compromise the next generation of cells. Yet the cell also has a daily requirement. It must selectively access specific microscopic segments of this code to produce the proteins that sustain its current metabolism. These two tasks face a significant physical bottleneck. Because DNA is anti-parallel, meaning its two strands run in opposite chemical directions, the machinery that unzips the helix faces a constant directional conflict. To function, the cell relies on a suite of specialized enzymes that resolve these geometric constraints without damaging the original genetic sequence. Replication begins at specific designated sites along the molecule, known as the origins of replication, or ORI. Here, an enzyme called helicase breaks the hydrogen bonds to unzip the double helix. Because opening the entire molecule at once would require a massive energy expenditure, the cell only exposes a small Y-shaped region called the replication fork. Notice the opposite polarities of the exposed strands. One runs 5' to 3', and the other 3' to 5'. This creates a mechanical limit. The primary builder, DNA-dependent DNA polymerase, is unidirectional. It can only catalyze the addition of new nucleotides in the 5' to 3' direction. On the template strand oriented 3' to 5', the polymerase moves smoothly toward the opening fork, creating a continuous leading strand. The opposite strand, however, presents a conflict. Because the template faces the 5' direction, the polymerase is essentially pointed away from the fork. It cannot build a single continuous line into the opening gap. To resolve this, the enzyme primase provides a workaround. It lays down short RNA primers that give the polymerase a starting point to build backward, away from the fork. The polymerase uses these primers to synthesize DNA in short disconnected segments, known as Okazaki fragments. Finally, DNA ligase seals these fragments into a single strand. This entire assembly line is powered by the building blocks themselves, the dNTPs, which act as both the physical substrate and the chemical energy source for the reaction. The cell utilizes this backstitching method on the lagging strand to ensure both sides of the master file are duplicated with perfect accuracy. While replication copies everything, transcription is far more selective, reading only one strand of a single gene to create RNA instructions. This restriction is necessary. If both DNA strands were transcribed, the cell would produce two complementary RNA molecules that would immediately bind together. This creates double-stranded RNA, which cannot be translated into proteins. The cell manages this through the transcription unit, a specific region of DNA defined by a promoter, a structural gene, and a terminator. Transcription begins when RNA polymerase associates with an initiation factor called sigma. This protein subunit allows the enzyme to recognize and bind specifically to the promoter sequence at the start of the gene. As the enzyme moves across the structural gene, it reads only the 3' to 5' template strand. This ensures that only one functional, single-stranded RNA molecule is synthesized. When the polymerase reaches the terminator sequence, it binds with a Rho factor. This interaction triggers the release of both the enzyme and the newly formed RNA strand. By isolating the transcription to a single strand, the cell prevents molecular interference and ensures its genetic signals remain clear. In eukaryotes, the freshly synthesized RNA is not yet functional. This transcript, called hnRNA, is interrupted by non-coding sequences known as introns. These introns must be removed through a process called splicing. Large molecular complexes, called spliceosomes, excise the introns and join the functional coding exons together in a precise order. To protect the strand from degradation, a methylguanosine cap is added to the 5' end, and a long sequence of adenylate residues, a poly-A tail, is attached to the 3' end. This multi-step verification and modification process ensures that only mature, error-free mRNA is exported from the nucleus for translation. These pathways show DNA's versatility. Replication uses both strands and DNA polymerase to create a permanent double-stranded copy of an organism's history. Transcription uses one strand and RNA polymerase to create a temporary single-stranded message for immediate cellular function. These two distinct mechanical objectives, one requiring total duplication and the other requiring selective extraction, are achieved using the same underlying genetic sequence. Through these mechanisms, the cell ensures that our genetic history is both preserved for the next generation and actively expressed to sustain life today.